Hey, AP Bio. So we want to just go over the last group of these free response questions. So this is topic one, free response group C. And so we've got this first question here, question one, which gives you this diagram of ice, and you've got the five questions that you're being asked to explain. So we're going to go right to those questions. So letter A, describe why hydrogen bonds form between the water molecules. That's 2 points there. And for each of these, you got 2 points. So if we jump to our PowerPoint, um, we'll get the answers to that. Okay. So question A, it's the slightly negatively charged oxygen atom, right? So in one water molecule, that's attracted to the slightly positive charged hydrogen atoms in another, so that you form those hydrogen bonds. So the response that you have should explain that the electronegativity of the oxygen pulls the electrons shared with each hydrogen atom within a single water molecule towards the oxygen, right? And so that's going to create those hydrogen bonds. Now, for part B, as water cools, we're going to see that the water molecules slow down. And so those hydrogen bonds become a little more stable. But with ice, those bonds cause the molecules to actually space out in a crystalline form. And so you actually create the ice lattice. So since liquid water molecules are at a greater temperature, you're going to see that the molecules move more rapidly, and they're going to always form and reform those hydrogen bonds. Now, ice is really interesting because it will actually reduce heat loss. It acts as an insulator, and that's because of the air spaces within the ice molecule itself, or the structure of the ice lattice. And so, water right underneath that ice is going to be actually warmer, and it's not likely to freeze all the way down to the bottom of the body of the water. Um, Water molecules are going to interact with hydrophilic, polar, and charged side chains of the amino acids. So that's the reason why you get those proteins to fold in a specific way. Hydrophobic side chains always tuck in to the middle towards the center of the protein, away from the surrounding water molecule. And for letter E, this mutation changes the sequence of amino acids. So that's going to affect the folding pattern of the protein since the side chains don't interact. And so what that will do is change the structure of the protein completely. Okay? All right. And then for question two, it focused on asking about this enzyme bromelain. And so we know that amino acids are the monomers, right? Amino acids are the monomers of which this enzyme is composed of. And what bromelain does is it enables the hydrolysis of either the protein or the polypeptide of the amino acid chain, and it breaks apart the peptide bonds holding the amino acids together, or that it simply just breaks the bonds between amino acids using water. You could even say it helps catalyze a hydrolysis reaction. That would work too. Now, if you change the pH, that's going to decrease the activity of the bromelain enzyme. Changes in pH affect the interactions between the R groups, and it changes the folding pattern of the enzyme, and thus it changes its shape and ultimately its function. Okay, so those are your other two free response questions. These are more similar to what you're going to see on the AP test. College Board was telling us that for the most part, they want you to be writing out and typing out the conceptual information on the AP exam. So you're not going to have graphs and diagrams to make, thankfully, but you are going to be asked to explain processes, justify your explanations, and make connections between different parts of the curriculum, like this question did. Okay. So again, going back to this one, I like this because it asks you to talk about water molecules and hydrogen bonding, but then it also brings in, okay, how does this impact what goes on with something like sickle cell anemia? Okay. And the reason why we have to predict the effect of this mutation on the structure is by doing so, talking about interactions between amino acids and hydrogen bonding. And then for question two, okay, again, they want you to talk about the monomer. They want you to talk about how does this work? Um, in terms of breaking the enzymes that cause browning. And then again, they want you to make a prediction here, okay, and provide reasoning. So this is important stuff. This is where they want to make connections with you guys. So these are the types of questions you'll see, okay? I hope that helps. Again, always reach out to us. Doc and I will always help you guys out if you have questions you're still not sure, okay? Good luck with everything this week, guys, and we'll see you soon. Bye.